This is Metabolism Part 3, and we're going to focus on the process after the glycolysis, which is TCA cycle. Here's a quick summary of glycolysis. It's conversion of 1 molecule of glucose into 2 molecules of pyruvate with a net gain of 2 molecules of ATP. And its overall equation is here, the glucose 2-ADP, 2 inorganic phosphate, and 2 NAD plus as coenzymes, converts into the 2 pyruvate to ATP, 2 NADH, 4 proton, as well as 2 H2Os. And there are 3 control points in glycolysis. And the first one is a hexokinase reaction, which happens in the step 1, and it is inhibited by its own product. And the second one is the phosphofructokinase in step 3, which is inhibited by ATP and citrate, and also can be activated by AMP, which serves as a cells or cellular starving signal. And the last one is the pyruvate kinase and its reaction in step 10, which can be inhibited by ATP and acetyl-CoA and can be activated by AMP and fructose 1,6-bisphosphate. And committing step in glycolysis is PFK reaction in step 3. And there are irreversible and essentially unidirectional reactions, which are hexokinase reaction, PFK reaction, phosphoglycerate kinase reaction, as well as pyruvate kinase reaction, which are essentially, most of them are these 3 control points. In addition to ATP, glycolysis also generates NADH and pyruvates, 2 molecules of a pyruvate, that must be dealt with later within this metabolic pathway. Under aerobic conditions, pyruvate, which is the final product of glycolysis, is processed through TCA cycle, which is a tricarboxylic acid cycle, also known as a citric acid cycle or Krebs cycle. So all these are the same thing. Basically for the complete oxidation of the pyruvate to the carbon dioxide with the production of additional several molecules of NADH and FADH2. And NADH is reoxidized back to NAD plus in the respiratory chain, which is the electron transport chain. In anaerobic conditions, the pyruvate is converted to other molecule by fermentation. So in case of yeast, it converts the pyruvate into ethanol, and other microorganisms and animal tissues, especially for hard-working muscles, the pyruvate is converted to lactic acid through the fermentation pathway. And in both cases, the pyruvate is reduced, and that makes the reoxidization of the NADH that is produced in the step 6 of the glycolysis that was done by the CAPDH reaction. And when you look at this first one, the yeast alcoholic fermentation, that has the 2-step. The first step is the pyruvate that is converted to the acetaldehyde with the carbon dioxide. And this reaction is catalyzed by the pyruvate decarboxylase. And the step 2 is that this acetaldehyde is converted to the ethanol. And this reaction is catalyzed by the alcohol dehydrogenase, or ADH, which is coupled with the NADH, NAD plus recycling. And this whole process for the alcohol fermentation is the basis for the beer and wine making. Another type of fermentation is lactic fermentation that happens in animal tissues under anaerobic conditions, such as rapidly contracting skeletal muscles. And in there, all oxygen molecules is consumed during strenuous exercise, so cells can no longer oxidize pyruvate via TCA cycle. So instead, pyruvate is reduced to lactate by lactate dehydrogenase, which is LDH, and this is the reaction here, where you can see the lactate and the lactic acid. And muscle cramps are due to the buildup of lactic acid, which leads to the acidification of the tissues. And this lactic fermentation is also known as step 11 of glycolysis, which is coupled to reoxidation of NADH to NAD plus recycling process. And this lactic acid is removed from the muscles by blood, and then the lactic acid is moved into the liver and resynthesized into glucose via gluconeogenesis. And the overall the reaction for this anaerobic glycolysis in muscle would be glucose plus 2-ADP and 2-PI, inorganic phosphate, is 2-lactate, 2-ATP, 2 water molecules, and 2 protons. So this diagram shows the different fates of pyruvate after glycolysis. So you can get fed into the this citric acid cycle or the TCA cycle to get oxidized into carbon dioxide as well as water molecules. And also oxidative phosphorylation happens involved in here for this process. The other two pathways, the fates are basically the fermentation. It can be the lactic fermentation, especially in the working muscles, and also alcoholic fermentation in yeast to generate ethanol. Now let's move on to the TCA cycle or citric acid cycle, which happens after the glycolysis through the metabolic pathway of glucose. So this TCA cycle is again tricarboxylic acid cycle, also known as Krebs cycle or the citric acid cycle. And this cycle allows much more complete oxidation of glucose, which means that there are going to be much more ATP is synthesized compared to what glycolysis alone would accomplish. So in the glycolysis, as you know, starting with the one molecule of glucose, it generates 2 pyruvate molecules as well as 2 ATPs, which releases only a fraction of the potential energy of glucose. But under aerobic conditions, pyruvate is converted into acetyl coenzyme A, which is acetyl-CoA, and then this acetyl-CoA enters the TCA cycle and is ultimately oxidized to carbon dioxide. TCA cycle takes place in a mitochondrion or mitochondria. There are huge and complex organelle in the cell that is responsible for the cellular respiration and generation of energy. And when you look at this structure of the mitochondria, you can see that there are 2 layers of the membrane, which are outer membrane and inner membrane. And there's an intermembrane space between those 2 membranes. And the inner membrane has a lot of folds called the cristae, and also the inside of the inner membrane there's a cytosolic area called the matrix. And there are many proteins stuck in the inner membrane who plays a critical role in the cellular respiration as well as oxidation. So electrons that are transferred onto NAD plus and FAD in this oxidative process in TCA cycle are ultimately used to reduce molecular oxygens through the metabolic pathway. And in this process of this electron transfer through electron transport chain in the mitochondria, the protons are pumped across the inner mitochondrial membrane into intermembrane space. So the gradient is created there, and it has both electrical and pH components in the gradient. And energy contained in this gradient of protons is used to make the ATP by the enzyme called ATP synthase, which is found in this inner mitochondrial membrane. And this process of ATP synthesis is coupled with the electron transport chain. And this process is known as oxidative phosphorylation, which we're going to discuss in detail in the next lecture. And complete cycle of oxidative phosphorylation can result in as much as 32 ATP molecules synthesized per each glucose molecules utilized. These are features of a TCA cycle. TCA cycle requires aerobic conditions, and TCA cycle is anabolic, meaning that it has both catabolic and anabolic roles. So it not only breaks down acetyl-CoA, but also provides several intermediates and precursors for biosynthetic reactions. And TCA cycle is a cyclic pathway, so it regenerates its starting intermediate, which is oxaloacetate. And TCA cycle oxidizes acyl groups to 2 carbon dioxide molecules, generating 3 NADH, 1 FADH2, and 1 GTP, which is later converted into ATP, and this GTP is energetically equivalent to ATP. So the overall reaction for this TCA cycle would be 3 NAD plus FAD, GDP, and acetyl-CoA with inorganic phosphate produces a 3 NADH, FADH2, GTP, 2 carbon dioxide, and coenzyme A. And TCA cycle is also a pathway for complete oxidation of amino acids and fatty acids, not just the glucose. And these two are also entered as the form of acetyl-CoA. But the pyruvate molecules from the glycolysis doesn't simply get into the TCA cycle, but it has to have a transition process. And that is done by the enzyme called the pyruvate dehydrogenase, or PDH complex, and its regulation. So the overall reaction for such transition from the glycolysis to the TCA cycle would be pyruvate plus coenzyme A plus NAD plus produces the acetyl-CoA, carbon dioxide, NADH, and the hydrogen cation. And its role is to prepare the pyruvate for the entry into the TCA cycle. But the import process is important because the pyruvate is basically in the cytosol or the cytoplasm, because glycolysis occurs in the cytosol. But the PDH complex reactions and TCA cycle, as well as the electron transport chain and the oxidative phosphorylation, everything occurs in the mitochondria. So there's a difference in the subcellular localization of the glycolysis and the TCA cycle. So in order for the pyruvate to get into the mitochondria, it uses the pyruvate H+, or the hydrogen cation import system. And the PDH complex, which is responsible for such transition, is found in the mitochondria of the eukaryotes and also present in bacteria such as E. coli. And their structures and function of E. coli PDH complexes are well known, well understood. And this PDH complex has a large complex. So it has 60 subunits in total. Pyruvate dehydrogenase, or PDH, is made of 3 kinds of enzymes which make the 3 different parts, E1, E2, and E3. And E1 consists of pyruvate dehydrogenase, which occurs as 12 dimers. So that makes 24 subunits in total. And E2 part comprises dihydrolypoyl transacetylase, which have 24 subunits, and that makes the scaffold of the cube of a PDH complex structure. So it defines the overall shape of that complex. And E3 consists of dihydrolypoyl dehydrogenase, which have 6 dimers. So that's going to be 12 subunits in total. So when you add all up here, that's going to be 60 subunits in one PDH complex. And this is an EM image, electron microscope images of the PDH complex, where you can see multiple enzymes get together to form this cubic shape. And there are also coenzymes for that different parts of the PDH complex. The coenzyme of E1 is thiamine pyrophosphate, or TPP, and coenzyme for E2 is lipoic acid in the form of lipamide, and coenzyme for E3 is FAD, which also requires magnesium ion. In addition to these coenzymes, loosely associated NAD plus and coenzyme A participate in the PDH complex reactions. Thus, PDH is a non-covalently associated multi-enzyme complex that carries out the oxidative decarboxylation of pyruvate to acetyl-CoA. This diagram shows how PDH complex works for transition from glycolysis to TCA cycle. So it has 5 steps from 1 to 5, and let's take a closer look at each step. So in step 1, E1 enzyme, which consists of pyruvate dehydrogenase, works with the coenzyme TPP, or it's also called as cofactor, decarboxylates pyruvate, so it strips out this acyl group and turn it into the carbon dioxide. And this acyl group is going to be transferred to TPP to form the hydroxyethyl TPP or HETPP. In step 2, the oxidation of this HETPP happens by the E2 to give this acyl dihydrolipoamide from lipoamide. And it basically gets this acyl group and forms a diester bond here. It also leads to the regeneration of TPP in E1. In step 3, which still happens in E2 and catalyzed by E2, there's a transfer of acyl group from the lipoamide to the coenzyme A to form the acyl-CoA. And also, it produces this reduced lipoamide on E2. Now we have acyl-CoA that is ready to enter the TCA cycle. But we still have this reduced form of the lipoamide that has to get back to its original form, which is SS form or oxidized form, which contains the disulfide bond here. Otherwise, this reaction cannot continue for more pyruvate molecules for processing to the acyl-CoA. So this step 4 and step 5 are basically to get this reduced form of the lipoamide to get back to the oxidized form of lipoamide for reusage. So in step 4, E3, which is a dihydrolipoyl dehydrogenase, which has the FAD prosteric group, reoxidizes the E2 lipoamide that is reduced form that contains SH and SH here into the SS form, which is oxidized form. And then this E3 itself is going to be reduced. And therefore, this E2 is regenerated in the oxidized form, which has this SS form. But now we must reoxidize this E3 again too to regenerate it into the back to the original state, which is oxidized form. So in step 5, reduced active sulfhydryls of E3 are reoxidized by its own prosthetic group FAD, which is then reduced into FADH2 that contains SH and SH here. And this FADH2 is then reoxidized back into FAD by this NAD+, which is not tightly bound to the enzyme here. And this NAD+ turns into NADH plus hydrogen cation. Therefore, this E3 is recycled back into its oxidized form. And as we discussed earlier, why is arsenic a poison? We talked about the similarity of its substrate here, of the certain enzymes. And when we look at this pathway here, this arsenic is specifically inactivates the lipoamide-containing enzymes such as E2 of the PDH complex, as well as alpha-ketoglutarate dehydrogenase of TCA cycle. Therefore, it brings the respiration to halt. There are several advantages of organizing PDH complex enzymes into a single complex. First, all enzymes are very close within the complex, and that makes their active sites being closed too, in which case the intermediates are going to be easily passed through from one to the next enzymes, so it achieves the substrate channeling there. So the diffusion is limited there, and the rate of reaction is going to be enhanced. And the other advantage is that these intermediates are not diffusing out of the PDH complex, so until the final product of a PDH complex reaction is made. So there's little to no chance for the side reaction to occur somewhere else rather than the PDH complex. And lastly, this complex allows a coordinated control of those 3 enzymes. Then why is this PDH complex controlled? it actually requires careful control because it's virtually irreversible reaction with its delta G naught as minus 33.5 kilojoule per mole. So it's pretty spontaneous reaction. And there is no other way in mammals to make acetyl-CoA from pyruvate other than this PDH complex reaction. And also making the acetyl-CoA just commits these carbon atoms to either going through TCA cycle or fatty acid synthesis, which is another fate of acetyl-CoA in the cytoplasm. So this PDH is a huge commitment, so it has to be really carefully controlled with a lot of controlling points or the regulation mechanisms. Then how is the PDH complex regulated? What are the mechanisms of that? The PDH complex is regulated largely by two different mechanisms here. First one's by the product inhibition. So the NADH and acetyl-CoA, which are the products of this PDH complex reaction, inhibits the PDH complex activity. So more specifically, NADH and the acetyl-CoA compete with NAD plus and coenzyme A on E3 and E2 respectively. And if E2 stays acetylated, that makes it cannot accept the acyl group from HETPP on E1 anymore. In turn, E1 is stuck in HETPP now and cannot decarboxylate the pyruvate anymore. And the second mechanism of the regulation for the PDH complex is by the reversible covalent modifications. And an example is going to be the phosphorylation of the E1 on specific serine, which inactivates this enzyme. So this only occurs in eukaryotes, but not in bacterial PDH complex. And it's accomplished by the PDH kinase, which phosphorylates the PDH complex, and it can be reversed by the PDH phosphatase. And PDH kinase inactivates E1 using the ATP as a donor of a phosphate group. So the phosphorylated form of the E1 is an inactive form of this enzyme. And PDH phosphatase removes the phosphate and reactivates the enzyme. And NADH and acetyl-CoA, in addition to their direct effects that we already explained here as the product inhibition, this it activates the PDH kinase too. And pyruvate and ADP inhibits the PDH kinase. This picture shows the TCA cycle or citric acid cycle from step 1 through the step 8 here. And let's discuss these individual steps in detail. So the step 1 is the step where the entry into the cycle and the reaction is catalyzed by an enzyme called the citrate synthase. And what it does is basically the condensation of acetyl-CoA and the oxaloacetate to get the citrate. And you can see the reaction right here too. And the hydrolysis of the thioester bond of acetyl-CoA, which has the high energy bond, drives this reaction forward with the delta G naught as minus 31.4 kilojoule per mole. So it's a negative value, so it's highly spontaneous. So this reaction is virtually irreversible. And because it's virtually irreversible, it should be highly regulated. So indeed, this reaction, especially the citrate synthase, is allosterically inhibited by the succinyl-CoA, which is the product of the step 5 in the TCA cycle. So it's an intracycle cycle regulator. And also, this step is inhibited by the NADH, which is the product made in 3 different steps in this TCA cycle. And this NADH works as an indicator of the energetic status of the cell. In step 2, citrate is isomerized into isocitrate, which is catalyzed by aconitase. And this aconitase isomerizes tertiary alcohol, which is citrate, into the secondary alcohol, which is isocitrate, which makes it easier to oxidize in the next step. And this is basically a two-step process involving the cis-aconitate intermediate. In here, the water molecule is abstracted from the citrate and then going to be added back as the H and OH here. But now it's going to be in the opposite position when you compare it with the citrate. And this reaction, interestingly, the delta G naught is positive 6.7 kilojoule per mole. So it's not a spontaneous reaction. But why does reaction go forward? It is because this reaction is pulled forward by a subsequent exergonic reactions, which is catalyzed by isocitrate dehydrogenase that has the delta G naught value as minus 8.4 kilojoule per mole. In step 3, they're going to be oxidative decarboxylation of isocitrate to form the alpha-ketoglutarate. And that is the first oxidative reaction of TCA cycle, and that reaction is catalyzed by isocitrate dehydrogenase. So this reaction is also a two-step reaction. For first step, there's an oxidation of a hydroxyl group at C2 to form the oxalosuccinate coupled with the reduction of NAD plus into the NADH and the hydrogen cation here. And the next step will be the decarboxylation of the oxalosuccinate that produces the alpha-ketoglutarate and the carbon dioxide. And this oxalosuccinate intermediate is unstable and easily decarboxylated to the alpha-ketoglutarate. So since this is a pretty unstable stage, this will happen pretty spontaneously. And indeed, the delta G naught is minus 8.4 kilojoule per mole. Therefore, there's a sufficient exergonic process or the energy to pull the aconitase reaction, which was the previous reaction in step 2, going forward. And NADH that was generated here later enters into the electron transport chain and is used in oxidative phosphorylation. And this process, the reaction driven by isocitrate dehydrogenase, is allosterically regulated. So ADP and NAD plus activates the enzyme, as opposed to ATP and NADH inhibits the enzyme. And actually, the reaction senses this NAD plus and NADH ratio and the ADP and ATP ratios, which are the indicative of a metabolic status of the cell. So the high ratio of these will mean that it's a green light for this reaction, and the reaction will go forward. In step 4, there is an oxidative decarboxylation of alpha-ketoglutarate, and this is the second oxidative reaction of the TCA cycle. So it produces the succinyl-CoA and the carbon dioxide coupled with the reduction of NAD plus into the NADH. And this reaction is catalyzed by an enzyme called alpha-ketoglutarate dehydrogenase. And this is also a large multi-enzyme complex which has a similar structure and the function to the PDH complex. And it acts through the same mechanism as the PDH complex works. And this enzyme is composed of these three components here, which are the alpha-ketoglutarate dehydrogenase, dihydrolipoyl transsuccinylase, and the dihydrolipoyl dehydrogenase, which is identical to that of the PDH complex. And this enzyme requires a similar set of cofactors as the PDH complex, such as magnesium ion, TPP, lipoic acid, as well as FAD. And this reaction produces the carbon dioxide, diester product, and the NADH with the delta G naught value as minus 33.5 kilojoule per mole. So it's negative value, meaning that it's the spontaneous reaction. And this reaction can be regulated positively by AMP and negatively by NADH and succinyl-CoA. In step 5, there is a conversion of succinyl-CoA to succinate with the production of GTP, and this is the only substrate-level phosphorylation step in the TCA cycle. This reaction is catalyzed by succinyl-CoA synthetase, and the high-energy intermediate succinyl-CoA is used to generate the GTP directly, and this GTP is also a high-energy phosphate. Thus, a substrate provides the energy for the phosphorylation rather than the events linked with the electron transport chain for the respiration. And this is the why it is called as a substrate level phosphorylation. The energy for the hydrolysis of a thioester bond in the succinyl-CoA, which is minus 33.4 kilojoule per mole as its delta G naught value, drives the reaction of synthesis of GTP, which requires the 30.5 kilojoule per mole. So it's enough energy that it can generate from this hydrolysis of diester bond. Therefore, the net delta G naught would be the sum of these two values, which is minus 2.9 kilojoule per mole. And GTP then exchanges the terminal phosphoryl group with the ADP to produce ATP, and GTP goes back to the GDP in this case. And this reaction is catalyzed by nucleoside diphosphate kinase. So here's the summary of the first 5 steps of the TCA cycle. First, it introduces the 2-carbon acyl group into TCA cycle as an acyl-CoA, and then it is condensed with the oxaloacetate, and then liberates 2 carbon dioxide and also generates 2 NADH and 1 ATP, which is generated from the GTP, and it ends up being the succinate. And remaining steps basically finish the oxidation and complete the cycle by regenerating oxaloacetate for the next cycle. In step 6, there's an oxidation of succinate to fumarate with the reduction of FAD to FADH2. And this reaction is catalyzed by succinate dehydrogenase, which is the only enzyme of TCA cycle that is membrane bound. It's especially localized in the mitochondrial inner membrane instead of being soluble in the matrix as all other TCA cycle enzymes are located. And this enzyme is also part of electron transport chain. You can easily imagine that because it's stuck in the inner mitochondrial membrane. And it reduces the FAD rather than NAD plus at this step because it is not sufficiently exergonic to reduce NAD plus. But still, this reaction is enough to reduce FAD. This FAD is a stronger oxidizing agent, so it is easier for it to accept electrons than for the NAD+. In step 7, fumarate is hydrated in a stereospecific manner to produce L-malate. And it involves the transaddition of the element of water, which are the H and OH group here, across the double bond here, which used to be the double bond, but it's broken now, to get this H and OH groups. And this reaction is catalyzed by fumarase, and it has the stereospecificity because there's only L-malate is produced rather than D-malate or the racemic mixture. In step 8, there's an oxidation of L-malate to return to oxaloacetate with the reduction of NAD plus to NADH by the enzyme called malate dehydrogenase. And this reaction is highly endergonic with its delta G naught value as 29.7 kJ per mole. And that results from the very low concentration of oxaloacetate in mitochondria, which is a product of this reaction. And how do we know that the concentration of oxaloacetate is low? We can start it from this equation where it relates the delta G naught with the KEQ. So that's specifically delta G naught equals minus RT ln KEQ. And in order for this delta G naught to be positive, ln KEQ must be negative. And therefore, this KEQ should be smaller than 1 because it's in the ln, the logarithm. And this KEQ is the ratio of the concentration of the products and the concentration of reactants. So this products reactants concentration ratio should be smaller than 1. And since this delta G naught value is quite high, this ratio should be much smaller than 1. So that means the product's concentration, which is an oxaloacetate concentration, is going to be very low. Then why doesn't this endergonic reaction go forward? It is because it's immediately followed by the highly exergonic reaction in step 1, where the condensation of the oxaloacetate and coenzyme A, acetyl-CoA happens. And that's delta G naught value is minus 31.4 kJ per mole, which is even when you add these two delta G naught values, that's gonna be ended up negative. So they can still have enough energy to pull this reaction go forward. As always, the coupling endergonic reaction to an exergonic reaction through common intermediates will pull the reaction go forward. Energetic balance of the TCA cycle as a whole will be with the delta G naught per 1 pass as minus 40 kJ per mole. So it's energetically favorable. And this is a summary for the PDH complex and TCA cycle reactions. So for the PDH complex reaction, 1 pyruvate molecules and the coenzyme A and NAD plus gets the acetyl-CoA, NADH, carbon dioxide, and the proton. And TCA cycle starts with the acetyl-CoA that was from the PDH complex reaction, and the 3 NAD plus FAD-GDP inorganic phosphate, the 2 water molecules to get the 2 carbon dioxide, coenzyme A, so it recovered back to that, and then 3 NADH, FADH2, and 3 protons and GTP. And remember that there are 2 pyruvate molecules gets into this PDH complex reaction as well as TCA cycles. So that's going to be 2 times of these molecules that are actually happening from 1 glucose molecule. So anyway, the overall reaction will be the addition of these 2 reactions. You can cancel out the one that is redundant on the other side to get the pyruvate for NAD plus FAD, GDP, PI, 2 water molecules to get the 3 carbon dioxide, 4 NADH, FADH2, 4 protons and GTP. And again, it can be multiplied by 2 when you want to consider the starting with just 1 glucose molecule. So this completes the catabolism of glucose that entered the glycolysis, and all 6 carbon atoms from glucose have been completely oxidized and liberated as carbon dioxide. In total, that's going to be 6, 2 by the PDH complex and 4 carbon dioxide molecules by the TCA cycle. So let's consider the energetic balance calculated in ATP equivalent. So basically from the glycolysis, there's a 2 ATP that is generated, and also through the TCA cycle, there's another 2 GTP that is generated, which is converted to the 2 ATP. In addition to that, let's assume that the 1 NADH is equivalent to 2.5 ATP, and 1 FADH2 is equivalent to 1.5 ATP, which occur later in the electron transport chain and oxidative phosphorylation, which we will discuss in the next lecture. So the full accounting of energy in the form of ATP extracted from the one molecule of glucose entering the glycolysis would be 32 ATP. That's a lot of energy. So again, during the completion of the glucose oxidation, the cells can recover energy sufficient to make up to 32 ATP per glucose molecule. But most of the released energy is not immediately used for ATP synthesis as in the substrate level phosphorylation, but it's rather stored in the form of reduced coenzymes such as NADH and FADH2. And complete oxidation of glucose can be written as a sum of two half reactions. Here, one is the oxidation of glucose, and the other is reduction of molecular oxygen.